Hello, everybody. I think we're ready to get started. Welcome to Diversigen's webinar, Behind the Data, with Dan Knights and Joe Petrosino. My name is Lisa Gamwell. I'm a product manager at Diversigen, and I'm going to be moderating this discussion and live Q&A. So with us today, we have Dr. Dan Knights and Joe Petrosino. Together, they have over 35 years of combined experience in the microbiome field and have authored over 260 peer-reviewed publications. In addition to their very productive academic careers, they were both instrumental in founding microbiome sequencing and analysis companies, and these two companies recently joined forces under the name Diversigen. They have a lot of insight and experience to share, I think you're all in for a treat today. So for the webinar today, we asked Dan and Joe to discuss three broad topics, and these are longitudinal studies done with shotgun metagenomics, novel microbiome analysis techniques, and ways to ensure insightful data outcomes. So they're going to discuss those three topics, and then we're going to have a live Q&A. If you have any questions at any time, please use the Q&A button, and we'll keep an eye on those. So I'm going to turn things over to Dan and Joe. Dan, I think you're up first.
Okay, well, thanks, Lisa, for the introduction. It was very kind of you. And so, I'm really happy to be able to talk about some of these aspects of microbiome analysis with Joe. I think there are going to be some interesting overlaps between the things that we're finding in different studies that will be informative. And what I'm going to talk about is really the big picture is using shotgun sequencing and longitudinal studies to get a lot more power out of microbiome science. And I'm going to focus in on this biological question as kind of an example case. Is diet the main driver of human gut microbiome variation? And there actually is some conflicting evidence about this. So, there are a number of studies showing that diet has a huge effect, that trumps genetics. When you look at some animal studies, but when you do dietary interventions in people, even if it's a very extreme dietary intervention, people generally tend to stay in their own little microbiome cloud. So, it's not actually clear what the right answer to this question is, and we'll try to get to the bottom of it. The data I'm going to be talking about is this very dense longitudinal shotgun metagenomics data that was collected by Abby Johnson, who is a postdoc in my lab. And Abby's she has a PhD in nutrition. She also knows how to code, and she's a registered dietitian. So she's able to bring a lot of interesting thoughts about the diet side of this, which we'll get to in a minute. But first, let's look at this microbiome data. So what you're looking at is 34 people sampled every day for 17 days, and we did shotgun sequencing on the stool microbiome. And then each of these boxes is one of the people, and then you can see the different colors are the different species. So you can see their microbiome actually varying over time throughout the study. And I love to just look at this because there's so many different things you can learn. Like, for one thing, people obviously have their own fingerprint. If you choose any one person, there's no one who's an exact twin. And in fact, they're ordered so that people who are similar are close together. Some people are more outliers than others. You can see those 3 Prevotella carriers over on the right. That red color is Prevotella. But overall, everyone has something unique about them in their microbiome. You can also see that some people are more variable than others. And for example, like this, zooming in on one here, this is Abby's microbiome. She gave me permission to show this. She had the most stable microbiome of anybody in the study, which she was very proud of, being a registered dietitian. And this is a whole new feature of the microbiome that we can begin to use in clinical studies, which is the stability. Now, of course, we're talking about diet, so we also collected a full dietary recall on every subject throughout the whole period. So we asked them to track what they'd eaten in the previous 24 hours before each sample. And that's what I'm showing on the bottom. And so now, because of the longitudinal nature of this, we have this very rich set of analyses we can do, looking at both the between person variation, but also the variation within a person over time. You might have noticed these two pink subjects here. They had something very interesting happen to them. According to Abby, it turned out that they were computer scientists. And that means that they were drinking only Soylent the entire time. So they hadn't really announced this to us before we started the study, but it provided kind of an interesting sub-analysis, because if you look at their microbiomes, they did not converge. These are two people actually who were cohabitating, who had almost identical diets for 17 days, and their microbiomes maintained all of their individuality. They also did not stabilize. So it shows that although diet may have a role in microbiome variation, this gives us a hint that there's definitely a lot of other sources of variation. So the first question we're going to try to understand is variation between people. So on the left side, I've got a principal coordinates analysis of the beta diversity between every pair of samples. And you can see it's colored by subject. Each person has their own microbiome cloud. Some people are more variable, some people are less variable, some people are kind of more outliers. And what we wanted to know is, if we look at the distance between the center of any two people's clouds in the microbiome space, does that correlate with the distance between their clouds in the diet space? And so on the right side is, I'm showing the same thing, but for diet. So that's the first thing we're going to look at. We're going to just take the average microbiome over the whole study, so get rid of all that fluctuation over time, and the average dietary intake throughout the whole study, and find out if those pairwise distances are correlated. And to do this, we started off by using just the 50 to 100 macro and micronutrients that are tracked in all of the foods, because everybody's eating slightly different foods. So this is a very simple way to compare them. So we projected all the foods into nutrient space, and we took just the average nutrients for each person throughout the study. And that really didn't work. There was no correspondence between the nutrients and the microbiome, no significant correspondence. So we thought about this a little bit, and we realized, this actually makes sense, because all the nutrients that we track, these are the things that you see on the nutrition labels when you buy something at the grocery store, are not largely the nutrients that the microbes care about. There is tons of other stuff in our foods that we're not tracking here. Lots of complex carbohydrates, different types of sugars, different types of fibers, flavoring agents, preservatives, and these are present in all foods, even whole foods, that we just have no information about. So Abby derived this approach where she realized that we could actually project all of the foods onto a tree, so that similar foods would be close to each other on the tree. And that allowed us to use methods from the microbiome field that use trees to inform the statistics. And so one thing we could do is say, okay, let's say we're comparing my diet to your diet. If you ate spinach and I ate kale, well, those are not identical, but they might be more similar, with more similar effects on the microbiome than a third person who ate an egg. So this allows us to share information from closely related foods. And when we combined the food, the microbiome variation between people with the variation in what foods people were choosing compared on that food tree, we tried to line these up. So we're looking just at average microbiome and average foods. And we found that they actually lined up quite well. So although the nutrients failed to explain between person microbiome variation. The food choices explained it significantly. And this was only when we were using the food tree to share information about closely related foods. So this is part of the equation. And in fact, we were able to measure how much of this distance between people is explained in microbiome space, is explained by their food choices. And it was about 20%, 19.4%. So this is half of the problem. And we know that there's this whole other component, which is the variation within a person. So now we're going to turn to within person variation. If we look at, say, the variation of a person inside their own cloud over time, does their movement in their own diet cloud predict their movement in their own microbiome cloud? Or another way to say that is, if you eat similar foods, one day, will your microbiome the next day be similar to what it would look like if you'd eaten those same foods later on? So we took the same approach here. Now we're looking at just one subject at a time. So this is a principal coordinates analysis of one subject's microbiome cloud. And same thing with their diet. And the diet is made using this food tree approach. And we did the same approach where we try to overlay them on top of each other and see how well they line up. This is called Procrustes analysis. And then we get a p-value out of it. And we can actually visualize this over time by putting time on the horizontal axis and then showing the first principal coordinate on the vertical axis. So if there were no relationship between what you recently ate and what your microbiome looks like today, then these two, blue and pink dot or blue and purple dot trends would not be tracking each other. They'd just be kind of all over the place. But instead, for this particular subject, we see that they're kind of moving around together. And in fact, it turned out that when we did this analysis, almost all of the subjects had a significant correspondence between what they'd recently eaten and their microbiome today. And on average, their food choices explain 24.4% of their microbiome variation. So this is the other component. And when you add these together, we have foods explaining 20% of the between person microbiome variation, and then 24% of the within person variation. That's almost 50%. So the answer is foods explain 45, maybe, it's probably a little more than that because we could have better methods for measuring food, which is a whole other topic. Foods explain, maybe around 50% of your microbiome variation. And then there's other factors that are explaining the rest of it. So the answer is, it is probably the largest driver, but it's not dominating necessarily what your microbiome looks like. Now, the other thing that we were able to do, because we had this longitudinal data, is ask whether there were associations between certain foods and certain microbes in each person. So, we're not looking cross-sectionally, which most diet studies have done to date, diet microbiome studies have done to date, where we're saying, do people who eat this particular type of food tend to have more of this particular microbe? Those studies have not found strong effects, generally speaking. We're actually looking at the personalized diet microbiome interactions. So, for example, this subject 35 here, when they ate more cakes, cookies, and bars, they tended to have more Roseburia annulinivorans over the next day or two. So this is their own personalized diet microbiome interaction. And when we did this, we found many significant associations across people, dozens, but very few were conserved across all people. So here's one example that was generalized where we see that across multiple people, there's a positive association between cakes, cookies, bars, and Roseburia inulinivorans. This actually kind of makes sense. I mean, it's biologically plausible. If you look at the name of this bug, inulinivorans, this is a microbe that ferments inulin. And that's a common bulking agent and fiber additive in a lot of cakes, cookies, and bars. So it could be that this is actually a real relationship that's conserved across people. And in this case, if we have enough subjects, we might see it showing up in the cross-sectional data as well. But there are other features, such as this dark green vegetables versus Bacteroides species 116, or pork consumption versus Bacteroides oolacea plenus, where the signal is different in different people. In some people, it's a positive relationship, and some people, it's a negative relationship. So in these cases, you would not expect to see a signal, even with a very large study, in a cross-sectional analysis. You have to do the longitudinal sequencing to find out about this personalization. So that's another cool thing that comes out of being able to do longitudinal sampling. Some other things, I mentioned stability. We actually can use stability as a trait. So we measured stability basically by the inverse of the self-to-self distance. So like how much is your microbiome moving from day to day, relative to itself. And we found that microbiome stability was actually highly correlated with the diversity of food choices people were making. And this is a new finding, and it was quite a strong signal. A correlation of 0.5 is pretty high for biological analysis like this. So something's going on there. The other thing we were able to do is actually find out how many samples we needed longitudinally to detect some of the signals that we found. And this is actually going back to the between person analysis. Remember I told you the correlation, the correlation between people was about 20%. So 20% of microbiome variation between people was explained by the diet variation between people. And that was when we averaged their microbiome across the whole study, and we averaged their diet across the whole study. And what I've done here is a little experiment where I took the average diet and locked that in for each person. But then I tried doing the same analysis with only one microbiome sample instead of taking the average. And the signal went way down. Here we only get, on average, about 15.5% of the microbiome variation explained by diet. And this is because we're subject to all of that longitudinal variation within a person. That's adding noise to this test. Once we get up to 2, just having 2 consecutive samples makes a huge difference. And then the signal continues to go up until we get to 5, 6, right around 7 samples, 6 or 7 samples is where we're not really getting any statistical improvement in the signal. So what this tells me is that even in studies where you're not interested in a longitudinal signal, you will gain a lot of power if you can just collect a few consecutive microbiome samples and average them. And I think this scares a lot of people because they think, well, there's already, so much burden asking a patient to collect microbiome. We really don't want to increase patient burden here. But we found that it's actually, once people kind of get into the habit of collecting stool samples each day, it's not really an issue. In fact, having them start collecting and then stop during an intervention and then collect again at the end, they might be more likely to miss samples or forget, less likely to be compliant than if you just had them sample the whole time. And you can always just freeze the samples and not sequence them. So, this is a really, really important finding that wasn't really in the paper we published on that study. Something that I found recently, you really need this longitudinal sampling to increase power. So with that, I'm going to hand it over to Joe.
So thank you, Dan. And so what I'll continue with is our use of 16S and WGS, shotgun metagenomic sequencing, to look at a couple of projects that we've been reviewing with clinical cohorts to look at the association of the microbiome with type 1 diabetes and cancer. So for the past 7 years, we've had the pleasure to work with clinical collaborators from around the world to study the microbiome of developing infants in type 1 diabetes. So the TEDdy cohort, which stands for the Environmental Determinants of Diabetes in the Young, looks specifically at children from birth to 15 years of age and has collected samples from over 8,000 children at a monthly and quarterly basis to look at a variety of clinical biomarkers as they associate with disease onset. So this is a prospective cohort. These children are selected by having a genetic susceptibility for type 1 diabetes. And among the studies that they've done, they've decided to look at the microbiome in these developing infants and children as they become, some of them become type 1 diabetic. And so in an investigation led by Chris Stewart published in 2018, we wanted to characterize the microbiome of these children from birth to 5 years of age, looking at stool samples that were collected monthly. So gut microbiome, or proxy for the gut, if you will, during this period of time when the microbiome is changing rapidly as it is, and then with the mission to try to look for signatures or biomarkers that could characterize this developing microbiome and then use those biomarkers as a potential tool to look for any abnormalities in those who become autoimmune positive or go on to become type 1 diabetic. And so with that preamble, what you're looking at right now are over 13,000 stools collected from 800 children who were in a nested case control match within the cohort, meaning that 400 of these children were going to either are type 1 diabetic or have antibodies that are progressing towards type 1 diabetes and are matched with a control who is either who is age matched, gender matched, and geographically located in the same countries. So either Finland, Germany, or Sweden, or in Washington State, Colorado, or Florida. So these stool samples are collected, and then we analyze them, and we sequence them by 16S sequencing in this particular figure, and then use Dirichlet multinomial mixtures clustering to identify samples that resembled one another to put them into what we call community state types. And so we can see here 10 community state types moving from left to right, and they're numbered at the top. And these samples, again, are categorized by the microbes that are present within them. And we can see that the top 25 or so organisms by the genus, again, afforded by 16S sequencing, are listed here on the right. So we found that there were 10 clusters that could easily be delineated using DMM clustering. If we look to the right, we look at the diversity within these clusters, and they've been grouped as such that the Shannon diversity measurement, so the abundance, so the number of different organisms that are present and their evenness and distribution are measured and can be seen to be increasing across this cohort of children. If you take the same samples, so exact same samples, and sequence them by whole genome shotgun sequencing, the same cohort could be segregated into 18 different clusters or community state types. And the reason for this is because you have, as you're aware, a lower level of resolution or higher level of resolution, getting to a lower level of taxonomy, namely the species. And so by speciating the collection of samples, we can get finer distinction of clustering. And so you can see that we were able to identify 18 different clusters using the same DMM methodology. And when grouping these clusters, you can see that we can scale them again based on their Shannon diversity in the lower right. And so you may say, well, what will you do with that next? And so what we looked at following generating these clusters was to identify how individual children progress through these clusters over time. namely how their microbiota in their guts evolved from 3 months of age to 46 months in this particular figure, through each of the clusters as they were accumulating organisms that were colonizing their microbiomes. So we know, again, within the first 2 to 3 years of life, you're rapidly accumulating organisms from your family, your environment, your food that you're eating, the changes in the food that you're eating. And so this is reflected now using community state types as a biomarker over a longitudinal period of time. So this is a way to consider microbiome data in a longitudinal study. And so what we can find is that within 3 to 6 months or 7 to 10 months, every child, so all 800 children, all 13,000 plus samples fit into one of two clusters, cluster 1. And then from there, the children followed different paths. And so each cluster, and this is using the 60NEST data, is represented by a spear. And the path taken to the next cluster is represented by a line. And the breadth of the line and the color of the line, as you can see on the right, the transition frequency, indicates how many children passed from one point to the next. And so looking through this, you can see that eventually almost all children ended up in clusters 7, 8, 9, or 10. Few children ended up in 7, but most ended up 8, 9, or 10. And we can identify a developmental phase, a transitional phase, and a stable phase where the microbes pretty much more or less at this point in time did not change as robustly as it did in the earlier transitional and developmental phases. How could this be used potentially for tracking a child in these early years of life by their microbiome? Well, if you think about infant and toddler doctor's appointments, we're constantly measuring them for progress, right? So we're looking at head circumference, we're looking at body length, body weight. And so these types of tools could be used, these types of maps, if you will, of transition of the microbiome could be used to make sure that your child is progressing on a normal, at a normal rate in terms of their gut microbiome development. If we saw, for example, as we start to do this with other diseases or with other afflictions, we'll start to see that some of these paths may not progress past the transitional phase or may not get to cluster 7, 8, 9, or 10. They may level off at cluster 4 or 5, and then we can say, oh, there may be an issue here. So this is one way that we can use 60NEST data and WGS data in longitudinal studies to be able to map the microbiome and use it as a biomarker for illness or health. Another consideration is, well, do we need to start, once we have all these data, do we really need to keep sequencing everything every single time we go to a doctor's visit? Because it gets kind of, as sequencing gets lower and lower in cost, it still is not quite as inexpensive, say, as PCR, and it's not as fast as potentially as PCR. So can we use other tools that we have for, say, detecting infectious diseases and leverage them for detecting key commensal organisms that could represent, say, a healthy or not healthy microbiome? And the answer is for sure. If we look at the individual clusters now in the WGS data set, we can see that there are certain taxa that seem to be key indicators or key biomarkers for a particular state. So for example, in cluster 1 with the WGS data, we see that there's an enormous amount of Bifidobacterium breve that's much more abundant in this particular cluster than it is represented in any others. In cluster 6, you can see it's at a high level, but with respect to other taxa that are present, it's much more abundant in 1. Similarly, in other clusters shown here with circles, there are taxa that can be associated either alone or in combination that would be signatures of that particular cluster. And when you do quantitative PCR, you can start to measure the relative abundance or measure the direct abundance relative to one another and be able to say, with PCR, this is a microbiome sample from, say, cluster 18 or cluster 12. And so we are evolving biomarkers and tools into diagnostic tools that can be then used to measure the health of the microbiome at a given period of time. And we're using, again, infants and development as a proxy, but this can be used with any cohort, any case control comparisons. Switching gears briefly, I have one slide on this, but there's been a number of studies and, of course, a number of advancements in oncology and immuno-oncology. In one paper that we recently published with Florencia McAllister at MD Anderson this past year, she was interested in looking at the microbiome of long-term and short-term survival after surgery for treating people with pancreatic cancer. And then she used a cohort of, and this is now looking at the microbiome of the tumor. So the tumor microenvironment, which has become a hot topic lately, looking at the microbes that are present there and noting that we have to use 16S data at the moment for looking at these types of samples, because if you did whole genome shotgun sequencing of these samples, you would get a ton of host DNA that would swamp out the microbial signature. So everything you're looking at here is done through 16S sequencing. And what she did was to look at the microbiota in the tumors of pancreatic tumors resected at MD Anderson and at Johns Hopkins as a validation cohort. And in figure B, using a left-C analysis, so looking at associations with specific outcomes, we identified specific taxa that were associated with long-term survival, indicated in red, and those that were associated with short-term survival, indicated in green. So you can see the taxa in this cladogram that are identified here in the key. If you look at the same taxa now, looking at an LDA analysis, you can identify taxa specifically that are associated with long-term survival and short-term survival, with having a significant impact over 4 in terms of an LDA score. So with this, you can start to identify in the tumors microbes that are associated with long-term survival that can be profiled after surgery, because you'll have the tissue accessible for sequencing, and then be able to start to make clinical decisions as to whether or not what needs to be done perhaps to help take a short-term survival prognosis and make it into a long-term survival prognosis. And that's what their group is doing right now. And so some of the taxa that were associated, again, now in a heat map format, so those identified by LDA are now being identified through heat map. So Pseudoxanthomonas, Saccharopolyspora, and Streptomyces all seem to be associated with long-term survival. So it brings us to the question, when is 16S ribosomal DNA sequencing better? So as we've just indicated in the last slide, so samples of high host contamination, so tissues, epithelial cells, those coming from microbes exposed to cell culture. There are sometimes times or examples where microbes are in enough abundance that you can get enough of a signal to do whole genome shotgun and not be completely swamped out by the host. But more often, especially if you're using biopsies and other types of epithelial samples, you're going to end up with high host contamination, and you could just use the 16S ribosomal DNA fingerprint, targeting that with PCR primers to get information about the microbiome of those samples. Another time when 16S is great to have on hand is when there's a sample where one taxa is highly dominant. So if you did whole genome shotgun sequencing, you'd get 99% of one specific organism, and depending how deeply you're sequencing the sample by WGS, you may not see the other taxa or very much of the other taxa that are present. And so having 16S to go deeper into a sample at a lower cost can sometimes be a better consideration. And cost considerations, even though Diversigen offers the ability to provide shallow whole genome shotgun sequencing, you really need to consider the question that you want to answer, and then how much money you have to spend, because if there are limitations, you could always do a tiered approach where you can start with the 16S protocol on all samples, and then triage that larger sample set into a subset of samples, say 10 or 20 to 25%, for whole genome shotgun sequencing to understand what's going on in the samples that are of most interest. So we're about to jump into our discussion, but just to highlight the recap, longitudinal data enables personalized microbiome analysis. So we heard from Dan about the personalized diet microbiome, looking at developmental patterns is exemplified in the TEDdy cohort, and looking at long-term survival biomarkers. But these are just 3 of many, many, many projects that I know Dan and I and Diversigen have been associated with. Repeated sampling increases power for discovery. You are your own best control is sort of the mantra that we live by. And so longitudinal studies really increase the power and the types of questions you're able to answer. Shotgun metagenomics provides higher taxonomic and functional resolution, but we do still see times where 16S is still appropriate for certain applications. And so with that, I'm happy to open the meeting for further discussion.
Okay, we're just going to get the video up from everybody and we'll dive into the questions. So a reminder, you can ask your questions using the Q&A at the bottom of the screen. We've got a couple already that we can get started with. So this first one is for Dan, and it's a question about the fiber nutrient category. Why do you think fiber nutrient category didn't have discernible effects on the subject's microbiome?
Well, that's a great question. I think one very plausible explanation is that, you know, when we say fiber, we're actually referring to hundreds of different complex polysaccharides, and we just have no information about those for the vast majority of the foods that we're eating. You know, even if you look at like a banana or a strawberry, it has a very different set of fibers. Even if you look at a ripe banana versus a non-ripe banana, it has a different set of fibers. So, you know, at some very basic fundamental level, obviously it's just biochemistry or physics. You know, this fiber substrate docks with this enzyme and gets metabolized. But when we try to lump all the fibers together, you know, I don't actually think we really should be expecting to see an association.
Cool, thanks, Dan. We've got one for Joe. This is about the tumor microbiome profiling. So beyond the prognostic utility of tumor microbiome profiling, is there any potential for the data to direct therapeutic approaches?
Sure, and I think that's actually the number one goal for a number of the groups looking at the tumor microenvironment. The idea is, as we've seen already in immunotherapy in the gut microbiome, that bacteria associated with tumors could produce signals that would allow recruit cells, such as immune cells, that would fight the cancer directly towards the target and actually help, whether it's alone or in combination with other therapies, be able to better target a particular tumor.
I've got a question that was submitted at registration, and it's kind of along the same lines, but this one is for Dan. Let me just find it. Do you think the microbiome could be engineered depending on external interventions like diet with predictable outcomes? What would be some challenges in doing that?
Absolutely. That's one of that's sort of like the holy grail. You know,
Yeah.
your microbiome is here at point A. You want to get to point B. What do you do? And one of the things we found looking longitudinally is that you can actually predict where someone's going to be within their own microbiome cloud based on, you know, the previous few days, you know, with reasonable accuracy. But that's very different from trying to get someone into a whole other cloud. And that has been very hard to do. So it seems as though, you know, like if you look at the Soylent subjects in our study who drank only Soylent, this meal replacement beverage, they hardly budged out of their own microbiome kind of configuration. And I think if we're gonna talk about actually transforming someone's microbiome to a new state, we're gonna have to do something either very targeted with, you know, a particular substrate that we know is going to go in and feed a particular microbe that's present, and that may need to be personalized, or very blunt, like let's give them broad spectrum antibiotics for a short period, which will just tamp down the current microbes, and then, you know, understanding who's there, we can start to feed certain members of them, or even seed with a few new members, like a probiotic approach, and feed those to try to get it to grow up into, you know, a new configuration. And it's gonna, in reality, probably be, you know, a combination of those depending on what the target is or what the kind of transformation is that we're looking for.
Very cool. All right, this one's for Joe. It's about the MD Anderson tumor study. Antibiotic administration, peri-surgical interventions, pretty common. Would that change the microenvironment? And did you take that into account?
Yeah, no, for sure. And so I think the key for any clinical study is you definitely need to be in close collaboration with the investigators, the clinicians who are handling the patients, handling the samples, collecting the metadata, because those factors, among others, can be very confounding when it comes to downstream analyses. And so with this particular cohort, the patient group that was engaged, both at MD Anderson and at Hopkins, typically we're undergoing the same types of regimens and interventions for their cancer, and those that sort of fell off in terms of their treatment, in terms of the sort of that normalization of their treatment, were excluded from the analysis. That being said, sometimes you can't control for all that, and so you do the best you can with the data that you have. And every once in a while, there are still signals that will appear above the noise that you can follow up on in models in the lab.
Awesome, thanks. We're getting quite a few questions about sample size. So I'm just going to try and summarize a couple of them. So things like, what's the best method for determining sample size for a cross-sectional versus a longitudinal study? How many samples should you include per donor? That sort of thing. So one was answered in text, but I'm wondering if you could both comment on how you determine appropriate sample size for your studies.
It's a tricky one. I'm going to punt to Dan first and go off I'll go off on longitudinal studies.
That's fair.
Yeah, sure. I mean, like obviously in any new area, it's hard to determine sample size because you don't know what the effect size is in your particular like disease or variable of interest. So the most kind of straightforward approach is to find a comparable study or a disease that you think will have sort of a comparable magnitude of effect on the microbiome. And then you can either, find out, get those data, find out by subsampling what sample size was really needed to detect an effect in that study. Or you can use kind of more traditional epidemiology stats to figure out, based on that effect size, what's the sample size that you need. Now, that's for cross-sectional. I think in terms of number of samples per subject, I'd have to know, are you talking about a longitudinal study where you're looking for some kind of longitudinal signal? In that case, more is better for sure. I mean, dense, like at least daily, at least weekly, if not daily. But if you're doing a cross-sectional study, you still should get some consecutive or repeat samples per subject. And that helps you overcome all the noise in the kind of within person variation. So I would say, in my own studies, I'm looking to get 3, if I can, consecutive samples per time point in a cross-sectional study to overcome that variability.
Okay, and this is a follow-up question. Do you average those 3 samples or however many you take? How do you handle those?
You can, yeah, that's again the most straightforward way to do it. You can use models that incorporate repeated sampling. Not every statistical test has kind of an easy way to handle repeat sampling, and so it's also acceptable to just average them. And as I showed, in the slides, like you really get a boost in the power even doing something simple like that.
Okay, awesome. Joe, did you want to comment on sample size and power?
Yeah, so I would add that, especially working with clinical cohorts, that there are going to be limitations with, especially if you're engaging in patient groups or groups that where sample collection is not going to be readily available, or groups that are distributed across a large geographic region that are not coming into a clinic, for example. And so it's best, it's easy to say, it sort of sounds dismissive, but every study has its unique characteristics that are going to influence your sample collection rate. So I would say, in addition to the points that Dan made, which are fundamentally how you should approach any study, know the interventions, know the patient cohort, know the exacerbation states, how many flares a patient typically gets in this particular cohort, making sure that if you're, so for, speaking specifically, obviously, the human subjects right now, but if there's interventions in mice, you want samples before and after treatment points and things like that. So general tenets of sample collection should be held. And we always go 2 to 3 samples at baseline, and then had one of those samples be at time 0, and then Dan's more is better, but in lieu of that, hitting before and after either disease exacerbations or treatment administration is the best.
Okay, so more is better. That's what I'm hearing from you guys.
Yeah, I wish I had a better answer for that, but
that's okay. You guys you're on the cutting edge. You're working on it. So a lot of people are thinking about variables. How do you control them? What information should you collect? So maybe you guys can comment on what kind of variables and metadata you capture beyond what you might have discussed. So some things people are wondering about, like seasonal and external temperature changes, age, having a pet, and specific to the microbiome tumor analysis, what sort of controls do you use, especially thinking about contamination? So they're wondering about how do I make sure I've accounted for everything?
That's a whole webinar in itself.
Yeah, I'll make a note next time. That's what we'll talk about.
So I think in terms of what other metadata to collect, when we looked at the TEDdy metadata, so again, you're looking at kids at a time when the microbiome was being assembled, and we went and looked at, and the special thing about the TEDdy cohort is that in addition to the samples being collected, so many more samples than what we were analyzing just from a microbiome perspective, but the parents were collecting diaries on a monthly basis that they were turning in with daily information regarding diet, psychosocial stressors, pets, siblings, vaccinations, antibiotics, food, and all that great stuff. And so we took all that data and married it to the microbiome data changes that we were seeing, and looked at what was influencing changes in those initial months of life. And the things that came out were the top 3 things or 4 things were things that you'd expect. So breastfeeding versus not breastfeeding, and the date that solid food was introduced. So the date that solid food was introduced had a bigger weight than breastfeeding, so as you probably would imagine. So there's a transition phase, and so as soon as you saw solid food, things changed, even if you were still breastfeeding. I mentioned antibiotics, siblings, so brothers and sisters that touch the child, for example, and then furry pets. So having a pet also had the biggest impact on changes in the microbiome of those infants. But to the point that, outside of that, sometimes you're not going to be able to collect every single point of data, because again, you have to work with your cohort, or you're not going to get the samples you need. You'll lose engagement. So of course, diet, antibiotics, having samples collected in a uniform manner. So this is a great sort of plug for DNA Genotech, because having an easy to use sample collection method that could be transported readily without having to worry about temperature is ideal. And then same time of day is another factor. So we were telling, when possible, collect the first sample every day, so that you're sort of starting to level out the contribution of the diet that may have those sporadic changes that could happen through the day.
Yeah, Dan, did you see any specific metadata? Like kind of similar question.
Yeah, pretty similar things. I mean, like even in adults, smoker status shows up, recent medication history for sure, proton pump inhibitors and things like that. Even over-the-counter medicines can affect the microbiome. I think the other, a really big one that tends to show up very consistently is geography. And that's confounded obviously with many other factors that are influencing the microbiome, but I think it supports this idea that when you're designing a study, it's much easier to control for those things in the sampling frame. So if you can only sample in this city, then you're going to isolate the effects of geography, out of the study and be able to focus on whatever your intervention is. rather than having like many different cities and trying to control for that
Right.
post hoc in the statistical analysis. So when possible, try to control for as many of those confounders upstream through your study inclusion and exclusion criteria.
Okay.
And that will increase your power.
So what I'm hearing is assume everything impacts the microbiome and control as much as you can, or capture the data if you can't control for it. Is that a fair summary, more or less?
I think so. I mean,
Yeah.
again, if you're going to put it into a large bucket, for sure. But
Yeah.
you're always going to be fighting the dynamic of your cohort. I didn't answer one element about the controls for cancer. So
Yeah.
with any low biomass project, you are going to run into situations where potentially contaminants can start to emerge or show up. Most of the time, the signal from a sample that actually has something in it, no matter how small, will swamp out the KICS contaminants if you're controlling the best the best you can for those elements in terms of reducing exposure to DNA and so forth. We always sequence blanks or try to sequence blanks, and if they pass, like, so for example, if there's not enough DNA or RNA in a particular sample, then the library won't work or the amplification won't work, and you won't get a signal, and then you'll know that what you're seeing above that is typically something you can work with. But when you do see something in the blanks, you depending on the level of contamination, you either throw out the experiment and start over if it's really bad, or you use that and keep in mind that if those same taxa show up as differentiators in your analysis, then you need to really scrutinize that if you're really going to say that the same organism that is showing up in your kit actually has an impact on health and disease. Usually those organisms are very different and don't weigh in those types of analyses.
Okay, cool. So we have only a couple minutes left. We've been getting a lot of questions, both submitted before the webinar and during the webinar, about multiomics. So one question is for Dan. Do you think the analysis approach you used in your diet and microbiome study, could it accommodate a third level of data? So could you add metabolomics data sets on top of that?
Absolutely. And that's one of the key next directions, because as I said, at the very basic level, like it's all physics, right? And if we know the structure of all of the chemical constituents that are taking place in these reactions, then we should be able to measure those effects in a very deterministic way. So yeah, I mean, the whole tree structure, it might apply to something more specific in terms of chemical structure of metabolites, of secondary metabolites, fibers, and things like that. It's not something that we've had the opportunity to try out yet, but I think it's a very good idea.
Cool. So kind of along the same vein, and this one's a jump ball, either of you can answer this one. What are some best approaches to obtain functional or metabolic insights from microbiome data? So I think they're referring to sequencing data. What do you guys think about that?
I mean, I think without necessarily plugging any one of them, I think there's a few tools. If you're using 16S data that you can compute function from those data, and Dan may have specific favorites that he likes to use. Tax for Fun, PyCrest is, there's a few others. From a metagenomic data sets, using, we've always, we've been working on our own tools, but using tools that Curtis Huttenhauer has generated have been worked well for us in the past. There's great room for growth in this area. So if you're an informatician, aspiring informatician in the microbiome arena, I think there's ways, an opportunity to improve upon those tools.
Awesome. Danny, anything you want to add to that one?
Well, I think, as Joe said, there is definitely still a lot of room for growth and improvement. And one particular area is in analysis of functional analysis of uncharacterized genes and proteins. And that's something, the majority of studies map to a database of known genes, which there are different tools for doing that with slight advantages and disadvantages. but being able to learn de novo about how new things we've never seen before are related to outcomes in a study is something that I think is especially kind of cutting edge.
Very cool. Okay. So we've got some 16S versus WGS questions. Oh, but you might have answered one by text. I will say that Joe and Dan, if you see any questions that you'd like to answer, just click answer live and it'll pop up that you'd like to answer it. But basically, how do you figure out when it'd be better to use WGS or 16S? How do you guys decide which one to use? There are some other questions that I can follow up with along the same vein.
I'll start, I guess. I think if money is not an option, then WGS is always almost always better, unless you're dealing with a particular tissue sample or samples with epithelial cells or other types of contaminants inside the sample where you just sequence everything that's there. You're going to sequence something else besides the microbiome. So classically, if you're scraping intestinal tissue from mice, or if you're collecting biopsy samples and sequencing those, WGS becomes a little bit more prohibitively difficult because of the amount of human DNA you get out. But in terms of information content, most of the time, almost all the time, WGS will get you what you need. There's probably some rare exceptions that where there's a very, very dominant taxa and many low abundant taxa where WGS would just swamp with the most dominant organism and leave you sort of grasping at straws for some of the others. But more often than not, WGS is better if you have the resources.
Yeah, that's pretty much exactly how I would look at it too. And I guess one thing I also consider is how relatively unknown the microbes are in the particular sample. So the less well known the microbiome, or the less well represented in the database, the deeper you'll have to sequence to find what's there, to be able to characterize it properly. So the cost trade-off becomes pretty prohibitive in cases where you want to do a really large study and say like ocean water or something like that, with a lot of unknown microbes. And that's where you can still get a lot of signal out of 16S, which does do a good job. It might not tell you what these unknown microbes are, but at least you can find out that there's something there that's associated with external factors.
Okay, cool. Thanks, guys. I'm just looking through. We have some pretty specific questions about your studies. There's one about the Predict1 study. Joe, sorry, so I'm just going to read it out. So that study showed the microbiome had a relatively small effect size when seeking to make personalized diet recommendations. Did I screw that up? This might be for Dan. Based upon your data showing that diet data accounted for 45% of variation, how do you think about looking at increasing the effect size that you're seeing? So is the cohort driven when looking at effect size, or does the predict data really imply the microbiome has really small prognostic power in personalized nutrition?
Well, I think, in terms of the prognostic power, we found that it may have very strong power for personalization. And this idea that, like, if you want to manipulate someone's microbiome with diet, you need to know their diet microbiome interaction network. That's very different from trying to understand at a broad scale, what's some food component that's going to affect everyone in the same way. Those seem to be fairly rare, whereas the personalization signal is reasonably strong. So I think really focusing in on longitudinal studies and looking for those personalized signals is where we're going to see the biggest prognosticating power.
Okay, very cool. Okay, so we still have lots of people online, and so if Dan and Joe are able to stay for a couple more minutes, we'll go through some more questions. Is that okay, guys?
Yeah.
Or do you need to go?
I have a couple minutes, sure.
Okay, So Joe, did you see a question that you wanted to answer? Or do you want me to throw one at you?
Oh, there was one on antibiotics and the microbiome that I may have
Mm-hmm.
botched up trying to answer. So,
Okay.
text, but simply put, what the general observation so far has been is that if you take one antibiotic course, your microbiome will generally return the way it was after that course is done. If you take two, it gets close, but maybe not quite to where it was before. If you take three concurrent or more antibiotic courses of typical length, so 7 to 10 days, three or more courses tend to drop you off in a place that's different from where you started. There's been a couple of studies, Roman study, and then I forget another author or another laboratory that sort of saw the same type of thing. It's been repeated in mice as well.
Okay, cool. This one is for either of you. What are some challenges associated with host microbiome integrative analysis? And what are some suggestions you might have to do that better?
Host microbiome integrative analysis?
Yeah, host genome and microbiome, I think, integrating the two data sets or looking at relationships between.
I mean, a lot of the integrative analyses or the systems analyses that people use right now involve a lot of making the data less complex, reducing the data to simple things that can be compared to more standard statistics. It's not the best thing in the world, obviously, when you're using such great data sets and you find yourself comparing very small numbers of relatively small numbers of features in the end when you do data reduction. There are some people that are doing some great work in doing integrative analysis with using bigger chunk of the data, particularly with human genomic sequencing and metagenomic sequencing at the same time.
Okay.
Something I can add to that, I think, is that there's a lot of disease etiology likely that's site specific. So, there's some interaction between host and microbial byproducts that's occurring at, for example, in Crohn's disease, at the location of a flare or in a particular part of the gut. So that's one of the big challenges still in human microbiome analysis, is that you have to have some way of assessing the microbiome activity at that site, and it's really challenging to do that with transcriptomics, on a biopsy, because there's so much host RNA there. You can do things though, like ex vivo culture, or use cultured human cell lines to do some of those experiments, like outside the body. And you can also do sampling of a biopsy and do 16S sequencing or amplicon sequencing at that site, as has been done in some cases successfully with tumors, colon cancer tumors.
Okay, cool. Joe, if you have time, there's a question for you about mothers and their babies. Is there a correlation of their microbiomes? Does it project over time?
Yeah, I was just about to answer that by text.
Yeah.
So there is definitely a correlation of microbiome with the mother and baby, especially right after birth. As you get further and further downstream in terms of time, diet, some of the other factors we talked about before will start to have a greater impact. And then, over time, you see the traditional, your microbiome is similar to your siblings, similar to your family. And then as you take concentric circles of your environment outside of your house, you start to see the similarity dissipate.
Okay. Another one about working with infant samples. So how do you make sure to correct for sampling depth and library size when you're looking at diversity, especially for low material samples like early neonates and infants?
Yeah, so that's a great question. And so one of them is cost. If you're doing 13,000 samples, you may end up having to be limited by how much money you can spend on each sample. Outside of that, looking at your rarefaction data, looking to see if you're sampling deeply enough to capture the majority of what's there is best. Sometimes those two features, how much money you can spend and how deeply you're able to sequence because of that, does not line up very well with the rarefaction curves. Fortunately, in our case with Teddy, we were targeting 1 gigabase of data per sample in the WGS data and 5,000 reads per sample for the 16S data, and that did a pretty good job most of the time. But you'll just have to kind of sample your data as you're looking at it to ensure that you sequence it deeply enough.
Okay, cool. How are we doing for time, guys? Are you?
I probably have to go to another
Okay.
meeting, unfortunately.
Okay. So I think we're going to wrap it up there then. Let me just pull up my final slide. Which is here. And sorry, bear with me. All right, so big thanks to everyone, the presenters, the people who submitted all their questions. It was such a great discussion. Like I said, there's a lot of questions that we didn't get to. We're going to get in touch with each of you and try and answer them one-to-one. If you want to, get right to it, you can send us an email at info@diversogen.com and we'll try and answer your questions for you. And if you're looking for more content like this, check out diversogen.com/resources. It's where we put our past microbiome webinars, blogs, and podcasts. So thanks again, Joe and Dan. This was amazing. And I hope everybody enjoys the rest of their day. See you later.
Thanks. You too.
Stay safe, everybody.
Bye.
Bye-bye.